Well, over the past 100 years or so, the Bible has been under a prolonged attack from its critics. And especially recently, it has been criticized by a number of scientists as it disagrees with the widely accepted viewpoint of the origin and the development of life. And that's one reason why many people have turned their back on the Bible and consider that it is irrelevant in the 21st century. And to many people in the outside world, the Bible is seen as a fable. It's seen as a primitive explanation of the world based on superstition. And in today's society, it is seen as something that is completely out of date. And those of us who believe in it are simply clinging to an old story from a previous age and an old-fashioned ideal that has no place in today's world. Science, on the other hand, offers an up-to-date, rational viewpoint. It is impartial and deals only with the facts, which makes it very difficult to argue against. Scientists make very compelling arguments and appear to destroy the teachings of the Bible about the origins of life and the universe. We trust scientists because they know what they're talking about. We use computers and mobile phones every day, and they work because scientists have designed them based on their understanding of how the world and how the universe behaves. When we drive our cars over bridges, we trust the fact that scientists and engineers understand the properties of materials, and they've calculated the stresses and the loading capacity to ensure the bridge will not collapse. We trust doctors and chemists who devise new medicines and vaccines for various diseases. And over the past century, scientists and engineers have achieved some truly remarkable things that would seem impossible at the start of some of our own lifetimes. And so we trust scientists. They work with facts and with logic, and it has got the human race a long way. So when scientists say the Bible is wrong, people trust them. And why not? They seem to be right about everything else. Well, not only that, but the vast majority of people in the world, do not have any scientific or engineering training. So we're not in a position to disagree with scientists. They're the ones with the knowledge and the expertise, and just look at what they have achieved. So people assume they must be right. Well, scientists work with facts. Strictly speaking, they work with evidence, information they have observed from experimentation. And from this evidence, they put together theories on how things work. And then they test those theories to see if they stand up to scrutiny. And the aim of this talk is to provide you with evidence and with facts, the same information that scientists have. And we'll have a look at the conclusions that scientists have come to, and you might like to consider if you agree with them, or in fact, if you might not be quite so ready to accept everything that scientists tell you, especially when it comes to the questions surrounding the origin of life. Now, what I'm not going to tell you is that the scientists are wrong and the Bible is right. In the spirit of Brother Lawrence's original talks, and this was originally a public talk that I put together, I'm just going to give you the evidence and then you can decide for yourself. Now, to do this, we do have to get quite technical, and hopefully it won't be so complicated that I'm going to bamboozle everybody. But some of it's quite complex and might be difficult to understand, but hopefully I'll make things as clear as possible. What I'm going to focus on today is the origin of life and the complexity of life, and see how that fits in with scientific theories. And even if you don't understand it all, the thing I hope you will appreciate is the sheer complexity of life. Well, scientists would have us believe that the universe started spontaneously in a big bang, and then over millions of years, life on our planet has evolved. And this is what scientists tell us based on the facts as they see them. And from this, they assert that the Bible's account of a creator must be wrong. But when and how did life start? When we talk about evolution, we're not talking about how life started. Evolution assumes life has already started, and it gradually changes from one thing and into another. But let's start with a more fundamental question. How did life start in the first place? Well, scientists have asked this question, and a lot of time and research has gone into attempting to answer it. The general consensus among scientists is that life somehow started spontaneously. That's the only option available if we remove the possibility that life was created by an almighty God. So scientists have tried to find the conditions that would have led to the spontaneous creation of life. And in the 1950s, two scientists, Stanley Miller and Harold Urey, working at the University of Chicago, came up with an experiment, a very famous experiment. It's called the Urey-Miller experiment, and I remember being taught about it when I was at school. Some of you may remember it yourself. This experiment tried to mimic the atmosphere of the early Earth, and it's still taught in schools today. And these two men said, what happens if we take a mixture of chemicals, a prebiotic soup, something that might have existed upon the Earth millions of years ago, assuming that's the age of the Earth, say water, hydrogen, methane, and ammonia. And what happens if we pass electricity through it to simulate lightning storms that might have been going on the Earth millions of years ago, assuming that's the age of the Earth? Could that produce life? And they set up an experiment to do just that. They set up some equipment which included two chambers that were joined by tubes in a circuit. And one chamber contained water, representing the ocean on the Earth, and that was heated. And the water vapor moved through the circuit into the other chamber, and then it was cooled by a condenser as it travelled back to the first chamber. And a trap allowed water and chemicals to collect, and sampling probes to extract material. And in the main chamber, they put the gases, ammonia, methane, and hydrogen. And of course, they would have had water vapor from the water. And then they discharged electrical sparks in the main chamber via a couple of electrodes, simulating lightning. And they set all this apparatus going, and they left it running for a couple of weeks. And after 2 weeks, they looked at the chemicals that were created. And to their surprise, they found that over 20 amino acids had been created. And amino acids, as you may have heard, are often referred to as the building blocks of life. Once amino acids are created, we can build proteins from them. And proteins are used to build cells and to control the processes that go on within the cells. Amino acids are essential for life. So scientists had struck gold. A very simple experiment had demonstrated how life could have spontaneously come about upon the Earth. And then since then, other scientists, even within the last 5-10 years or so, have repeated and elaborated on this experiment and produced other chemicals that are needed for life. Once we have life, we can then use evolution to explain how different forms of life developed. So there you have it. Life is not so difficult to create if the conditions are right, which they might have been on the Earth. And it doesn't need any intelligent intervention or design in order to come about. These were random reactions, and yet these building blocks that lead to life were created. Well, that's how the scientists like to portray it. And if that were the whole story, then I'd just pack up and we'd all go home. But let's just wind back a little and have a closer look at this experiment and the deductions that were made from the results. When Miller and Urey came up with this experiment, they made some assumptions. And all good scientists will state their assumptions when they write up the results of their experiments. And then their main assumption in this experiment was the composition of the atmosphere. They came up with these gases, hydrogen, water, methane, and ammonia, by guessing. They didn't know these were the gases that were around many years ago. In fact, no one knows what gases were around. Maybe they were the same gases that we have in the atmosphere now. But Miller and Urey knew that these gases were the most likely to generate the right kind of chemicals. These gases might produce amino acids. So instead of finding out what gases were on the Earth and how they might be used to make life, they worked in a backwards sort of way. They knew the answers they wanted to get, and they came up with an experiment that was most likely to produce those answers. Now, that's fair enough. That's okay. But we need to be very careful what deductions we then draw from an experiment that's been constructed like this. We cannot say, for example, that this proves the Earth was made up of these gases. The composition of the gases is still an assumption, an educated guess. Do we have any proof that the gases that made up the atmosphere of the Earth many years ago? No, we don't. Let's just have a look at the gases again. We've got hydrogen, we've got water vapor, ammonia, and methane. What gas is missing? Anybody know? Oxygen. I mean, it is there technically. It's in the water, but it's not oxygen gas, gaseous oxygen in its free state. So why was oxygen not included in the experiment? It's because oxygen in its free state, like we have in the atmosphere today, is like a poison to amino acids. Oxygen reacts with amino acids and it breaks them down. And if free oxygen were present, like we have in the atmosphere, then the amino acids produced that have been created would have been destroyed as quickly as they were created. So what has the scientific deduction been? Well, scientists assert that there must therefore have been no oxygen in the early atmosphere. Now, we've got to be careful here, because this is a deduction based on an answer that we want to get. It's not based on any observed scientific evidence about the Earth's early atmosphere. It's a deduction based on an assumption. Scientists believe that atmospheric oxygen was produced by plants millions of years after the start of life. Now, notice I said there, scientists believe, because that's what it is. It's not based on any scientific fact. Now, maybe that doesn't matter. So what if there was no oxygen? Well, if there was no oxygen, then that gives us a problem. We've probably all heard of the ozone layer. There is a layer of ozone in the atmosphere that protects us from ultraviolet rays from the sun. And without the ozone layer, the ultraviolet rays from the sun would destroy all life and the planet would be completely dead. So without ozone, there would be no life. Well, what is ozone? Anybody know? O3, that's right. It is oxygen. Most molecules we have in the atmosphere are in the form of 2 oxygen atoms, O2. Ozone are made from 3 oxygen atoms, O3. But just hold on a minute. We're told that there was no oxygen in the early atmosphere because life cannot form with oxygen present. But then we're told life cannot survive without oxygen being present. So life can't start with oxygen, it can't survive without it. How strange is that? We might argue this would indicate life could not come about spontaneously. Maybe it points to an intelligent creator. So how do scientists explain this? Well, we simply modify our theory. In order to be protected from the sun's harmful rays in the non-oxygen environment, perhaps life began under the sea, perhaps near the hydrothermal vents that are found at various points around the globe. And so these are seen as candidates for the origin of life. How the amino acids would have come about here is not known. Scientists haven't suggested any mechanism. Perhaps they were made under the sea, under the Earth's surface, and then injected into the sea. The only trouble is amino acids undergo a process in water, undergo a process called hydrolysis. And the oxygen atom in the water molecule prevents amino acids from linking together, and eventually they're broken down. And so over a relatively short period of time, amino acids would be, once again, completely destroyed. And also the temperature of these vents is very high, and organic chemicals are highly unstable at these temperatures. So the amino acids would just decompose at the temperatures that these vents are at. So let's just stop and think about the evidence that we have. Based on scientific facts, that's what scientists work from, based on the facts, life cannot have occurred naturally on the surface of the Earth if atmospheric oxygen was present. Life could not have survived on the Earth if atmospheric oxygen was not present. Life cannot have been created under the sea because water would have prevented amino acids from being preserved. The facts lead us to consider another possibility, that intelligent intervention is required in order to create life on the earth. And in fact, that's precisely what the Urey-Miller experiment showed. The textbooks don't always include all the details. When Urey and Miller set up their experiment, if they wanted to demonstrate there was a chance life could have started spontaneously, then what they should have done was set up the initial conditions for the experiment and then leave it to run its course. They could not interfere. If they interfered, then they were introducing intelligence into the process, and that would completely invalidate the purpose of the experiment. And what did they do? They interfered. They had to interfere. In the experiment, the amino acids were created in a special reservoir, which contained a trap door. And Miller and Urey extracted the amino acids as they were created and stored them in special vials. And why did they extract them? Because they knew they would not survive the conditions of the experiment. And furthermore, there's another piece of information that we need to be aware of. Amino acids are molecules that have a certain shape. But in fact, for any given amino acid, there are two shapes that it can have. Amino acids have handedness. One's left and one's right-handed. And if you look at my hands, they're made up of the same elements. I've got 4 fingers and a thumb on each. But they've got different shapes. And it doesn't matter how I turn my hands, even if I do that, they bend different ways. They are different shapes. No way I can make my left hand look like it's my right hand. And amino acids are the same. They have right and left-handedness. And all reactions that create amino acids create a mixture of left and right-handed amino acids. And unsurprisingly, the Urey-Miller experiment created a mixture of left and right-handed amino acids. But if we look at the amino acids in every living creature, the many trillions of cells that there are, the many gazillions of proteins, they are all left-handed. There's not a single right-handed amino acid. Now, interestingly, sugar molecules also have handedness, and if we look at all sugars that occur in living creatures, they're all right-handed. So once these amino acids have been created, some mechanism needs to be in place to separate and to extract the left-handed ones in order to produce the proteins that are required for life. And if a right-handed amino acid is used in a protein chain, then that protein is no good at all, and we'll see that in a moment. So what undirected, accidental process produces only left-handed amino acids? Well, there isn't one. Scientists have come up with suggestions of how in a reaction you might get a few more of one than the other, but they're only small imbalances. And don't forget, it must have come about accidentally, without intelligent intervention. Some might say that the evidence suggests an intelligent creator. It looks like some intelligent process has taken place to organize things. Things do not simply go from a state of disorder to a state of order. What does the Bible say? It says, in the beginning, God created. Now, I hope you're all keeping up, because so far we've been looking at the simple stuff. We've been looking at amino acids, the building blocks of life, and we've been looking at some of the problems that are faced with creating these chemicals in a random, undirected, accidental way. And we keep using this term, the building blocks of life. We're looking at the individual bricks that make up the building that we call life. And scientists say that these amino acids, they join together to form proteins, as if that were a straightforward process. We're going to stand back now and look at the bigger picture. We're not going to focus on just the bricks. We're going to stand back and look at the building. In fact, we're not going to look at the building. We're going to stand back and look at the city that has been created from these blocks. Let's assume that spontaneous creation has occurred. Let's assume all the problems we've just looked at are overcome and left-handed amino acids have been created. Now we need to join these amino acids together into proteins. And I'm going to show you how that's done. And to do this, we're going to have a look at the so-called simple cell. And sometimes when describing evolution, it's said that complex chemicals collected together to form a simple cell. And the impression given is that a cell is a collection of just a few chemicals that could work together and reproduce themselves. But the simple cell is far more complex than that. The cell is the basic unit of life. All living things are made up of cells. And plants and animals are made up of many different types of cells. And the human body contains more than 10 trillion cells, and there are about 200 different types of cell. And all cells have common features, whether they are animal cells like ours or single-celled organisms like the euglena that's in this picture. Euglenas live in fresh water, and in warm weather they're what forms the green scum on the surface of ponds. And a cell is alive. It is as alive as you or I are. It breathes, it takes in food, it gets rid of wastes, it grows and reproduces, and in time it dies. And this picture shows the main parts of a typical animal cell. And if you think of it like a house has got lots of separate rooms. It's got a kitchen for doing one particular type of job. It's got a living room where some other activity goes on. There's bedroom, bathroom, all these different parts of a house. So in the same way, a cell has got specialized areas that are partitioned off for separate uses. And these areas include the mitochondria, which produces the cell's energy. We've got the nucleus, which controls the cell's growth and its function and its reproduction. And in the nucleus is the DNA, the cell's source of information. We've got something called the lysosome, which is the cell's waste disposal unit. We've got the Golgi apparatus, used to store and transport proteins. And we've got something called the endoplasmic reticulum, which processes proteins. So even already we're saying it's not such a simple thing as a simple cell. Don't worry, I'm not going to test anybody after this, if you can remember any of these names. But there are more than 20 different sections in a cell. And the cell is continually making new proteins and getting rid of old ones. And we're going to have a look at the manufacturing and transport process for proteins. And we're going to follow the journey of a protein from the nucleus to the lysosome, which is a distance of about 0.002 millimeters. And the lysosome is the cell's waste disposal unit. Well, when the cell detects, and that's a throwaway comment there's a lot involved in that. When a cell detects that it needs more of this protein, the manufacturing process is set in motion. And every protein has the information for its manufacture stored in the nucleus in the form of DNA. And you're probably all familiar with the pictures of DNA, this double helix, and it's all folded up and stored in the nucleus. And the DNA contains like the blueprints for making every part of you. So within the DNA, there's a section which describes a bit of protein that might be in your skin, or another bit of protein that might be elsewhere. The DNA describes every single part of you. And if you want to create, say, a new piece of protein, you need to go along the DNA and find the section of DNA that describes that protein. And then you do something with it. And I'm going to show you what happens next. I'm just going to move around here. I'm going to see if I can use this microphone around here. Because I just want to be looking at the screen when this next bit comes up. So we're going to see this process of the creation of this protein. So here we've got a cell, and we're zooming into the nucleus of the cell, which is where the DNA is. And you see at the edge of the cell, there's got all these round holes. Those are security gates. And they let only certain things in and out of the nucleus. So we go through one of these gates. As we go in, we can see the DNA there. This is this famous double helix. And we want to make a particular protein from it. So we need to zoom along the DNA and find the section which describes our protein. And when we get there, there's a little molecular machine that unzips that little part, unzips that little section, which has our protein instructions in it. And another little molecular machine comes along and makes a copy just of that section. So it makes a copy of that little section of DNA. It actually makes like a mirror image of it. And that little section is called RNA. This little section is copied. And it puts a little security marker on that RNA. The RNA then goes to the nucleus wall, and there's a security gate. It scans that marker, and if it's happy with what's on that RNA, it opens up and lets it out. So out goes this RNA. This is the instructions for how we make the protein. The RNA then gets picked up by something called a ribosome. And a ribosome is like a factory for making proteins. And when it's in there, the ribosome then joins the amino acids together. Do you remember the amino acids we were talking about? So here are the amino acids on the bottom, and they're being joined together in a very specific sequence that's dictated by these bases along here in the RNA. And each base has got 3 different sort of letters, as they call them, sort of shapes. And that dictates exactly the order in which these amino acids get joined together to make what is called a polypeptide chain. And that's our protein. It's not a protein yet. It's still technically a polypeptide chain. Here it comes out here. It's not a protein until it's been folded. So it needs to be folded in a certain way. So it gets taken to the endoplasmic reticulum, where it's going to get folded. And this is where the handedness comes into play. If it's folded in a very specific way that's dictated by the shape of each amino acid. And if you had a right-handed amino acid in there, it would bend the wrong way. So it's got to be folded in exactly the right way. There's also more in there that if it gets folded in the wrong way, there's a mechanism for detecting this and unzipping it a bit and then rejigging it back in again so that it gets folded in the right way. So that then gets folded up, and it comes out and it's picked up by something called a vesicle, which is like a little transport mechanism. And it gets taken on to the next stage. So that's just been built folded in there it's built in a ribosome, folded in the endoplasmic reticulum. It now needs to be taken to the Golgi apparatus, which is like the post office, and then it's got to get taken from there to the lysosome. Oh, sorry, I'll just come back to that. So I've got another animation coming up. Now, it's a different style to the other one, but it picks up exactly where that last one finished off. So hopefully it won't distract you too much that it's a different kind of animation, but it's picking up exactly where the previous one finished off. As I say, the Golgi apparatus acts like a post office. You'll see that. So this is the vesicle coming away from the endoplasmic reticulum. And this vesicle actually you can see it's got 3 proteins inside it. It gets taken to the Golgi apparatus. And the Golgi apparatus, as I said, is like a post office. You'll see that in a second. And this vesicle merges with the Golgi apparatus and it delivers those proteins to it. And the proteins get a little sugar molecule added to them. And that's like an address label. It tells the cell where that bit of protein needs to go. So then as it's moving through the Golgi apparatus, all these proteins get sorted depending on where they're going. It's like the address gets looked at and they're sorted into different positions. And each protein is moved into different parts. So you can see they're just like a post office, really. And then there are these like security markers, which then latch on to the protein, these receptors latch onto the protein that needs to go to a particular place. It's detected by that sugar molecule, latches onto that protein, and that small part of the Golgi apparatus then buds off. It just comes off and forms a little parcel. Pop, there it goes. It zooms off, and then it heads off towards the lysosome, which is where the protein needs to go. This is the waste disposal unit of the cell. So off it goes. Now this vesicle also has a security marker on it. It can't join to the lysosome unless it's got the right security marker on it. So it gets there, delivers the protein. Protein gets delivered in, and it's reached its destination. But you can't use it just yet. The protein needs to be turned on, in effect. So it needs to be activated. So that happens when it gets into the destination, gets turned on, and now it can be used within the lysosome. And then that part of the lysosome buds off again and heads back to the Golgi apparatus to be recycled. Well, I don't know about you, brothers and sisters, but I find that absolutely remarkable. That goes on inside a cell. That's just one of the process. And this process takes place many billions of times in every cell in your body during your lifetime. So when we're told that amino acids are the building blocks of life, we can see that, in fact, amino acids are just the starting point of an incredibly complex process. And this is just one of many such processes that must take place regularly in all cells in order for life to continue. And when there's just one defect in this system, the results are tragic. There's something called eye cell disease, which is caused when there's just one defect in the process and the protein never reaches the correct destination. It's like a letter being delivered to the wrong address. And as a result, the cell cannot dispose of its waste products properly. And the terrible consequences are there on the screen. And this shows just how vital it is to have all of these complex systems in perfect working order before the cell can work. And no one has offered a plausible explanation of how these systems could have developed as required by evolution. In fact, scientists really struggle with the evolution of the cell. Here's a quote from one evolutionary biologist, a man called Ali Demirsoy, who is at least honest enough to say, I don't know. He says, one of the most difficult stages to be explained in evolution is to scientifically explain how organelles, so that's the different parts, the different sections of the cell, developed from these primitive creatures. No creature or group has yet been found with organelles of a simpler construction in any way, or which are more primitive. In other words, the organelles carried forward have developed just as they are. They have no simple or primitive forms. They don't know where they came from, which could be shortened to, we don't really know how it's happened. And we must remember not to confuse facts with theories and suppositions. The facts we've been looking at are that the cell is the basis of all life, and it is incredibly complex. It contains many irreducibly complex systems. And tragic results occur if just one element of the many complex systems does not work correctly. And no one has ever discovered a partially developed cell. And no one has explained how the cell has evolved. So it could be argued that the many irreducibly complex systems in the cell suggest a designer and a creator. And it's often assumed that all scientists believe in the spontaneous chance creation of life, followed by evolution. Well, just for your information, there is a website called Descent from Darwin, and it was set up by a group of scientists who do not accept the theory of evolution as a satisfactory explanation of the complexity of life on the Earth. Scientists from around the world can apply to add their names to this list on the website to show their disagreement. And the only criteria is that you must have a PhD in a scientific discipline or be a qualified medical doctor who is a professor of medicine. So you have to be quite sort of knowledgeable people, scientifically trained, just to get onto this list, to show that you disagree with Darwin or with evolution. And over the last few years, hundreds of scientists have added their names to the list. And despite the impression that we get about science and scientists, especially when it comes to the origin of life, there are some things that scientists just don't know. Now, I've got a video clip in a moment of one well-known evolutionary biologist commenting on the origins of life. And I used to just play it, assuming people knew who this man was, but some people don't know. Richard Dawkins, I don't know if many of you know and love him, but this is his explanation or commenting on the origins of life.
Well, how did it start?
Nobody knows how it started. We know the kind of event that it must have been. We know the sort of event that must have happened for the origin of life.
What was that?
It was the origin of the first self-replicating molecule.
Right, how did that happen?
I told you we don't know.
So you have no idea how it started?
Nor has anybody.
Nor has anyone else. What do you think is the possibility that intelligent design might turn out to be the answer to some issues in genetics or in evolution?
It could come about in the following way. It could be that at some earlier time, somewhere in the universe, a civilization evolved by probably some kind of Darwinian means to a very, very high level of technology and designed a form of life that they seeded onto perhaps this planet. Now, that is a possibility and an intriguing possibility. And I suppose it's possible that you might find evidence for that if you look at the details of biochemistry, molecular biology. You might find a signature of some sort of designer. And that designer could well be a higher intelligence from elsewhere in the universe.
So, there you go. I played that clip not to mock Richard Dawkins, but to highlight the fact that there are limits to what science can currently tell us, such as the origin of life, and that even the most devout anti-creationists might be prepared to consider that life on Earth might have been designed. We've seen how incredibly complicated life is, even in the simplest of cells. And this is in fact just what the Bible tells us. Psalm 139, I will praise you, for I am fearfully and wonderfully made. Marvelous are your works. So here are some points to think about. Firstly, we have seen that science has no answer to how life on Earth started. And evolution has not proven to be a satisfactory answer to the development of cellular biology. Secondly, we must remember that often people want to believe in evolution because creation is unacceptable. And thirdly, remember that when the facts are considered, science cannot rule out the possibility of a creator. Even evolutionary scientists say it is difficult to believe that our incredibly complex, organized world happened by chance. So, in conclusion, I would appeal to you, don't ever allow yourself to be deceived. Scientists make very bold statements and make it sound like they have all the answers about life and evolution. But dig a little further, and you'll find nothing could be further from the truth. The more scientists discover, the more questions you realize that they have. And as you have seen, they don't have a clue as to how life began or how it became so complex. We all have the God-given ability to understand these things. Wisdom and knowledge are not the special prerogative of people who consider themselves to be experts, whether they are scientists or theologians. And so don't take my word for it. Have a look at the complexity of life and what scientists have to say about it. I think the organization and incredible complexity of life cries out that there is a creator. The heavens declare the glory of God, and the firmament showeth his handiwork. Day unto day uttereth speech, and night unto night showeth knowledge. There is no speech nor language where their voice is not heard. He causeth the grass to grow, and the herb for the service of man, that he may bring forth fruit out of the earth. When we just stand back and look at the world around us, the visible things, the invisible things, when we look at the incredible creatures that inhabit the earth, whether it is the birds that cleave the air or the animals that hunt on the plains of Africa, or the microbes that live in the soil, and we consider the complexity at every level, even down to the smallest element of life within the cell. There is such beauty in the creation all around us. And we can but marvel at our Creator and praise his works, for we are indeed fearfully and wonderfully made.